Hey folks, today I want to start talking about the digestive system. We're going to do the overview stuff at the beginning, as far as the functions of the digestive system, as well as the microstructure of the alimentary canal, like overall. We'll see as we're doing individual organs, there are some modifications to the wall of the alimentary canal, but we'll talk about the generalities first, and then we'll talk about, basically getting the food from the mouth down to the stomach. Here are the SLOs that are going to be included in this today in case you want to pause. But you can refer to the other documents for help with this as it goes along. Okay, one of the things that I really like talking about the digestive system is that this is something where a lot of people have like a good amount of baseline knowledge. Even this would be true even if we hadn't already done a lab exam on it, on the digestive system already. Like most people have a certain amount of walking around knowledge of the main parts of the digestive system and basically what it does. But I want to make sure that we're all using the same language for everything, because I've found over the years that being picky about this helps to make things easier that we're communicating the idea that we actually think we're saying. So there are a few functions that might seem overly picky, but I want to make sure we're all on the same page. Especially, ingestion is pretty straightforward. It's getting the food into the mouth. We remember, I suppose I should define at the very beginning, that the alimentary canal is this pipe that goes from the mouth to the anus, and you can think of it as sort of a disassembly line that the food goes through in whatever time that it has. And the body evolution has allowed us to evolve a number of different systems that are going to try to get the food particles down to the smallest possible size so that the enzymes that we apply to it are going to have the highest amount of function. Now, warm-blooded animals, this is important for them because if without being able to get as much energy out of our food as possible, we're at a real disadvantage. Cold-blooded animals don't have the same constraints that mammals do, say. Anyway, once the food gets in, now your textbook refers to mechanical digestion and chemical digestion. I will usually talk about mechanical breakdown, which is decreasing the particle size of the food. So figure if you just took a big bite of a carrot. You gotta mash it up in your mouth for a while, and when it gets small enough to be comfortable to swallow, there are sensors in the mouth and the top of the pharynx that are going to kind of let you, aliquot little bits of food down as it's small enough to chew. I can tell you that people who study this kind of thing for a living have very particular terms, but I think that I want you all to adopt this mechanical digestion as a process which mainly happens in the mouth, but kind of continues when it gets down into the stomach as well. Propulsion happens throughout the alimentary canal because you got to move it from place to place. After the mouth and basically before it gets down to the rectum, it's all involuntary, so we're looking at smooth muscles for much of it, but getting it down the pharynx, to the top of the esophagus, is a process that's where it starts to become autonomic. Everything, but just the very ends of the pipe are the ones that are going to be voluntary. We'll talk a little bit about different kinds of propulsion in a minute. Now, chemical digestion, I tend to refer to this as like enzymatic digestion, because there's breakdown that happens in the stomach where acid is applied to the food, but to a large degree, what that stomach acid is doing is not really digesting the food as much as it's just decreasing the particle size even further. We know this from, if you look at, all right, this is going to be the gross example. You can kind of tell where the food is coming from based on what it looks like after you throw it up. If you vomit and you notice and you look at it, it still looks like food. You can still identify food items that you had at your last meal. Then it's coming out of your stomach. But the food that, if you continue to vomit and it starts coming out of the small intestine, the food is usually, first of all, it's going to look usually yellower or greener because of the bile that's mixed in. But also, it's like this slurry of liquidy stuff. You don't see real identifiable chunks of food anymore. So really what the stomach is doing is it's breaking down the food into small enough particles so that it can get worked on by enzymes. Now, so most of those enzymes are coming in the small intestine. That's also where most of the absorption of foodstuffs happens. And we see here on this figure that absorption is going to happen largely by blood vessels, but lymphatic vessels are also going to be important, particularly for the absorption of lipids. So I guess the main thing I want you to take home between the two types of digestion that are listed on this figure is that mechanical digestion, that's like, I always call it mechanical breakdown, and that means breaking down, decreasing the particle size of the food. Chemical digestion can include the stomach acid, but really the stomach acid is still just decreasing the particle size. So I usually refer to enzymatic digestion as what's happening. We were going to take, say, your starches and turning them into simple sugars, or taking full proteins and turning them into amino acids. So basically, the enzymes that do that digestion, those are the ones that are taking the food polymers and breaking them down into the monomers that make them up. Because that's how your digestive system functions. If you eat a hamburger, it doesn't mean that you suddenly have a whole lot more muscle because you ate a muscle where the patty was made out of where the burger was made out of muscle. What happens is your stomach will digest that burger patty, of cow muscle, assuming cow muscle, and it's going to break it down into the smallest amino acids, like those individual, breaks apart all those Legos so they get just down to the very single bricks, and then absorbs those monomers. And then it rebuilds new muscle, new proteins from those raw materials. Your body does not take fully formed proteins from another species, from another individual, and then absorb them completely. What it does is it breaks apart those biomolecules from your food into its smallest monomers, and then the body can do what it needs to with it. Going to use it as raw materials, or going to be able to snap it apart for energy. So that's largely the task of the small intestine. The large intestine, by that point, most of the digestion of our food has happened, and the great majority of the food has been absorbed by that point. Any usable food that's in the large intestine really doesn't make it into a very large degree. There's a very small amount of, actually food stuff getting absorbed there. Most of what gets absorbed in the large intestine is water. So this water, if we just, didn't pack up all the food, we didn't kind of pack up the food and extract the water out of it, we'd get dehydrated really, really quickly. We basically lose most of our fluid volume in the better part of a couple of days, which is why diarrheic diseases are such killers. Even though diarrhea is a joke with my kids, it's the kind of thing that it actually does kill people in lots of parts of the world. So anyway, what happens in the feces is that's the food that's either indigestible or stuff that you didn't have the time to absorb. So even though I would say we tend to group together urine and feces because they both end up in the toilet, but they represent really different things. The feces, that's the food that you didn't use. So all that corn and celery and pennies and ball bearings, anything that you ate but didn't get absorbed into your bloodstream, it basically didn't enter your body. On the other hand, the food that did get broken down to its simplest monomers and then sent to the cells and then metabolized by them, your body doesn't make a big patty for those cell wastes. Those cell wastes are small, right? Those cell wastes are things that we're going to be talking about when we're doing the renal system later. So what the urine is those are, it's water, but it's also breakdown products of metabolism. That's food you did use. But the feces, that's food that you didn't use. I'll talk about details of all of these as we go along, but I want to make sure we're okay with these terms. Within propulsion, there are a couple of terms that you need to be hip to. These are relatively simple, but I want to make sure that you own them. First of all, peristalsis is a term that we have as almost equivalent to movement in the digestive system, but peristalsis is strictly movement in the forward direction, movement toward the anus. Well, all right, I take that back. Reverse peristalsis is what causes vomiting, but the normal kind of forward peristalsis is what's going to move the food in this kind of inchworm kind of fashion from one part of the alimentary canal to one that happens a little later. Segmentation, on the other hand, is a little bit different. I sort of like this figure because it's almost like mashing up the food by kind of alternately squeezing segments of the pipe so that you end up mixing together adjacent packages of food. Here's another word you can have. The term bolus is a term that refers to like a swallow of food or individual like packet of food. We usually talk about boli when they're swallowed, but any little pocket of food like this, we can consider a bolus as well. So segmentation happens a lot in the small intestine. The stomach, because its muscle wall is more complicated, it does even more complicated movements. We just call that mixing. Okay, now I said before that a lot of the processes that happen with the digestive system are autonomic, or at least automatic, where we don't, once we start eating, it's kind of like we're not in charge anymore. So there are different kinds of reflexes that happen within the digestive system. I'll talk about what we call the long reflexes first. So basically, when you start thinking about food, you smell food, people start talking about food, these external stimuli will turn on particular parts of your brain, particular hypothalamus, and go, yeah, I could eat. So what these long reflexes are, is these are things that try to prepare the digestive system for wherever the food is going to be next. We'll talk about these in more detail as we go, but for example, if it's getting to be about lunchtime, I look at the clock and I say, oh, geez, it's almost lunchtime. And I say, actually, I'm kind of hungry. You feel your stomach will you might start to salivate a little bit. Your stomach might start to roll, but nothing's happening in your intestines yet, because basically what's happening is your brain is sort of preparing the more forward parts of your digestive system to get ready for that food to come in. So as the and then when the food's in the stomach, you're gonna feel full, so your brain's gonna feel sated, and your stomach movements are gonna slow down as well as it's kind of aliquoting the food out into the small intestine. But so when the food's in the stomach and getting mixed up, then the small intestine is gonna start revving up, and the pancreas and the liver are gonna start to rev up, and then after it's been kind of largely done in the small intestine, then it's gonna get moved over to the large intestine. So it's always preparing the gut's always preparing for where the food's gonna be next. On the other hand, your digestive system kind of lives in the now, meaning that it doesn't like see the future or anything. So years ago, I had a student who asked me, like, how come my brother has to go to the bathroom right after eating Thanksgiving dinner? Like, why doesn't he hold on to the food longer? And the thing is, the food that he's defecating out isn't the meal he just ate. What happens is, if there's a really big load of food that's made it into the stomach, a really large meal has the effect of sending forward in the digestive system the message. It's like, okay, everybody clear the decks because there's a big load coming. And so then what happens is basically all that other food, your body just goes, okay, just drops it, pushes it out, and then it's going to work on this bigger meal. So if you have like people who feel the urge to defecate after a large meal, that's not that food. That's not that you just ate. It's the stuff that's in front of it. And your body's just dealing with whatever work is being given in the moment. Anyway, so the long reflexes are the ones that are controlling, really large, distant parts of the digestive system, and they involve the brain. On the other hand, there are a ton of ganglia of nerves in what's called our enteric nervous system, the kind of gut-based nervous system, where there are chemoreceptors that don't feed to the brain either, and there are stretch receptors that are also going to be sensitive to where the food is. So these short reflexes are going to be doing things like this segmentation, where you need to know, like, all right, these little egg-shaped boli of food, there's stretch on the muscle wall there, so it'll cause to stretch little places where the food isn't. So like the short reflexes are doing inch by inch kind of communication in the digestive system, whereas the long reflexes, those are ones that are kind of directing the big picture, but the details are worked out by the short reflexes, by the intestinal nervous, by the enteric nervous system. There's actually a lot of super interesting literature about this, but it's kind of beyond what this class needs. But this is one of the brain always gets most of the news as far as neuroscience goes, but there's a lot of interesting research that's happening for the enteric nervous system as well. Okay, you've seen this in lab, but I want to make sure we kind of pack things in a little bit. Remember how your body builds pipes. There's always a lining, then there's muscle, then there's a connective tissue wrapping. In the case of the alimentary canal, if we start from the lumen, there's this innermost layer that we're going to call the mucosa. Now, it's called the mucosa because that's the outer body surface. I always tell people if there can be air next to it, it's an outer body surface. The mucosa is a surface that's going to have basically the entrance of ducts so that enzymes or mucus or or bile or pancreatic juice can get applied to the food, what's inside the gut. Another word we use for the food, because after it starts looking unrecognizable as a meal, we start calling it chyme, which is just a term that refers to the food plus whatever has been added to it through these various excretions. The mucosa has just deep to it a layer called the submucosa. Now the submucosa is like this additional layer between the and that's where I suppose if you look at the mucosa itself, there's a little lamina propria as well as a layer called the muscularis mucosae. The muscularis mucosae doesn't really move the food, but what it does is it kind of, I almost think of it the way that a horse will kind of ruffle its skin in order to shrug off flies. This is what allows the gut wall to kind of shrug off food so it doesn't adhere to it. So it kind of keeps the chyme moving. But externally to the mucosa is the submucosa. And what you can see living within the submucosa are a bunch of large multicellular glands. It's also where a lot of the blood vessels and the nerves and the lymphatic vessels that are going to service that part of the gut, they're all going to kind of travel through there in a plexus. So there are the various intrinsic nerve plexuses as well as the blood vessel and lymphatic plexuses as well. And again, a plexus is just a little network. External to the submucosa, the submucosa is the layer that's been added in the digestive system compared to our usual setup of pipes. External to the submucosa is the muscularis externa. Remember, the interna is it was basically they're referring to that's the opposite of the muscularis externa would be the muscularis mucosae. We remember from lab that there's always an outermost longitudinal layer of muscle, and then in 90 degrees to that, and a more an inner circular layer. We know that the stomach has 3 layers. I always remember that the longitudinal layer is always on the outside. That way I can remember what gets added onto it elsewhere. Then external to of course the muscularis externa is where most of that propulsion as well as segmentation is happening. That's where those are the muscles that are causing those movements to happen. External to that is the wrapping. Now the wrapping, all the parts of the digestive system have what's called a serosa, but this is but in the case of the esophagus, where there's no peritoneum around it, we call it the adventitia. That's kind of a fine point, but the serosa on the outside usually has an outer wrapping of the peritoneum on the outside. So I'll get into the peritoneum in a sec, but remember things like that greater omentum that we saw in the cat, or the mesentery, or the mesocolon. Those are all different, what we call extensions of the peritoneum, that are holding the abdominal organs in relative position. Speaking of the peritoneum, the text gets into, I'm not going to totally walk you through this diagram, but the main thing that I want you to take away about the peritoneum is that it's a series of serous membranes that are going to be kind of slinging the organs and holding them into place. There are what we call both dorsal mesenteries that attach the pipe into, or any other organs, to the back body wall. There are some places where there are ventral mesenteries as well, things like the falciform ligament that holds the liver in place. And in some cases, during development, at the bottom, this is showing like the kidney, basically, where the kidney starts off with this dorsal mesentery that holds it in place, but it kind of it migrates its way back behind the peritoneum. This is why you had to kind of pick out the kidney when you're dissecting the cat. It's hanging out behind the peritoneum as in the adult stage, just because of the little migrations of tissues that happen. We'll do more about that when we do the urinary system later. There are a number of different peritoneal extensions, but if you look at it, most of these are different dorsal ones. So, for example, if we work our way down, the lesser omentum, that's basically there between the liver and the stomach. It's what holds the kind of posterior to that is where the pancreas lives. The mesocolon is what holds the large intestine into place. The mesentery is what holds the small intestines in place. And we remember also before that there was a kind of this apron-like greater omentum that kind of flopped over down the front. That's that's a characteristic we also see in mammals. It's got a lot of fat in it because in addition to the subcutaneous fat, this is another place where mammals keep their fat. The visceral and parietal peritoneum that are labeled here, those are general terms that refer to all of them, but the extensions of the mesentery are really the two omenta, as well as the mesocolon and mesentery, and falciform ligament of the liver. Okay, so let's work our way down the pipe now. So, of course, this is going to happen as we're going through these lectures, where we're looking at organs that we've dealt with in other lectures. We're not going to get into a ton of detail. The main part of the organ, what happens with the oral cavity, is going to be mechanical breakdown. But there's some amount of enzymatic breakdown that happens there as well, because the salivary glands insert, the saliva, and saliva is mostly water. A little bit of mucus, but it's also got salivary amylase. Salivary because it comes from salivary glands. Salivary amylase is an enzyme that breaks down starches and turns them into simple sugars. There's an experiment you can do, actually, where if you take unsalted saltines, has to be unsalted. If you use the salted ones, you'll just taste the whole time. But if you chew up unsalted saltines and chew them up for a while, after a while they'll taste gross for a while, but then it'll start tasting sweet because the salivary amylase will be converting the starch that's in the cracker into sugars like glucose that your tongue will taste. So it takes a while. It takes about 3 to 5 minutes for most people if they end up trying that. I used to do that in class, but I stopped doing it, and of course I can't do it because I can't see you guys anyway this semester. But so the saliva is there largely for lubrication. That's the biggest part of it. But in most mammals, there's also salivary amylase. Some other mammals actually have growth hormone that can come out through the salivary glands. It's actually really interesting. So for dogs licking their wounds and stuff like that, it cleans it, but it also stimulates those cells to grow. Humans don't do that, but that's the thing that does happen in other mammals. And of course, the teeth are here as well, as far as what's going to be the main part of doing the mechanical breakdown, and the tongue participates in that. Here are a little bit about the teeth here. They are, of course, in humans, we know that they're kind of given different names. One of my favorite bits of information, and forgive me if I've told you this before, is we all know that the third molar that comes in later in life is called the wisdom tooth. But as it turns out, the other two molars also have names, and they're these totally Victorian names. So the first molar comes out, the 6 or 7, like it says here. That's called the school tooth, right? Because it comes in when you're in school. But the second molar, that's called the factory tooth, because by the time you're 12, you're working in the factory already, right? And then assuming you survive the factory, then the wisdom tooth comes in later. So these different kinds of teeth have particular shapes that are doing different kinds of breakdown. You have your spatula-like incisors, the canines, which are pretty reduced in humans, but they're larger in other mammals. as well as your premolars or the bicuspids, that are they're a little bit more for grinding, and then the molars whose whole job is grinding. This picture, I don't think this is in the current edition of your textbook, but I think it's cool because you can see this is a juvenile mouth. You can see where the teeth are labeled on the left side. You see like those permanent teeth that are coming in above the baby teeth, above the deciduous teeth that are going to be pushed out. In most cases, unless you had your baby teeth pulled out by a dentist, you'll probably notice when you gave a tooth to the tooth fairy, it didn't have any roots on it. That's because the root part of the tooth gets reabsorbed before the permanent teeth come in behind it. And actually, in this individual, if you look on the extreme lower right and left on the mandible, you can see like just a crown of those wisdom teeth. So you can still see. We would say that those are wisdom teeth that are still in the crypt at this point. A little bit about the structure of the teeth, just briefly. Looking at a simple kind of. Oh, by the way, this should be SLO. Hello, at the top of the figure. Most of what we see is what we call the crown. The teeth are covered by enamel. Enamel is actually the most mineralized tissue made by any vertebrate. It's meant to be hard, and it's meant to be able to withstand abrasion. The dentin that's below it is softer, sometimes will get exposed in aged people's teeth if they have a lot of wear, but for the most part, it's the enamel that's doing the job. If you or a family member has what are called receding gums, that's when the gum, like the gingiva, kind of recedes past the neck of the tooth. So what's being exposed there are that softer dentin and also more nerves. So people tend to have discomfort with like hot or cold foods if they have a lot of receding gums. And then what's not shown very well here, just because it's a 2D diagram, is the periodontal ligament. So the sockets that the teeth fit in are just called the alveolar processes. Now, what holds the tooth in place is this periodontal ligament, which is sort of 3D. It's almost like a tent that's connecting the root of the tooth to the root of the tooth to the bone itself. On the right, the little figure I threw in there, there's just a micrograph of a cross-section of the dentin and then the enamel. Enamel's really interesting too because it is built during development, right? So like when these permanent teeth are coming in, what has to happen first is the dentin gets laid down, and then on top of it there are cells called ameloblasts that lay down a layer of enamel on it. So each of those, if you look at the enamel side of that figure, you notice it almost looks like you see like a bunch of little lines kind of oriented like 30 degrees to the right. That's those are individual like lines of activity, almost like tree rings that show the job that the ameloblasts are doing. And the ameloblasts build like one or a couple of those layers per night. So really what we're looking at is like a clock for how long it takes the tooth to get built. Anyway, enough about that. When the food, basically where the food is headed after the mouth, is going to be the esophagus. The esophagus is where these layers of the alimentary canal are going to be like their most obvious, where there's a pretty thick mucosal layer because there's a lot of abrasion that happens there. There's a submucosa that kind of holds things in, to kind of spackle things in, and then the muscle. But there's not a ton of digestion that happens here. There are like no new enzymes that get introduced. Really, it just has the saliva and salivary amylase that came in the mouth. But this is just a transit place, to propel the food from the pharynx down into the stomach. So let's talk a little bit about swallowing, or what the big kids call deglutition. Basically what happens is, so there's this bolus of food that's in the mouth, and then when the food is soft enough, the individual particles are small enough, and the food's been sculpted so that it's not going to be too scrapey, what happens is the tongue will kind of move that food to the back of the mouth, and then pass it, and basically it'll cause the uvula to close off the nasopharynx, and then the food will get that bolus gets kind of squirted its way down. So if you compare these two figures, I should have lined them up better. It's here the epiglottis is up, Down here, the epiglottis has been bent. So that's preventing the food going down going down to the larynx like we'd expect. But then this is the point here when the bolus of food is like getting behind the cricoid cartilage there like it is, there comes a point up at the top here, there's this upper sphincter that's going to close down. At this point, this is where the peristalsis starts. So at this stage, the peristalsis is just beginning. And you can like try this at home, but don't choke when you do it. If you try to like guide food back really slowly in your mouth, you can like almost feel the beginning of those peristaltic waves, and then it'll just kind of grab the bottom of it and pull it along. But anyway, if once peristalsis is started here, it takes about 10 or 12 seconds, but it'll take a while for it to kind of move its way down to the stomach. And don't forget what it's passing behind. It's like passing behind the airways, it's passing behind the heart. There's this little hiatus in the diaphragm that where the esophagus is going to join up with the stomach. And then eventually, there's a lower sphincter. There's a lower opening here that I would call the gastroesophageal opening, only because I don't think it's a very real anatomical sphincter. It doesn't have much thickened up circular fibers. That's kind of a fine point. People will call it the gastroesophageal sphincter, but I don't think that's all the way correct. That's the main stuff I wanted to get through for this. Shoot up any questions to the discussion board, and I'd be happy to have any feedback. Thanks.